Hello, my name is Elektra Papadopoulos, and I work in the Division of Clinical Outcome Assessment in the FDA's Office of New Drugs. And our mission is to promote the development and implementation of patient-focused endpoints in medical product development. And today I'd like to discuss some of the key considerations in clinical outcome assessment. Of course, I'll be expressing my own views and not necessarily an official FDA position. Over the next 15 minutes or so, I'll discuss some of the regulatory considerations for clinical outcome assessments for use in medical product development to assess the benefit of therapeutics. And following that, I'll describe some of the ways that stakeholders can engage with FDA in development or selection of COAs. So now I'll talk a little bit about the role of COAs in assessing clinical benefit. Before we go into the details of measure development and selection, let's step back and think about the broader context of what we're trying to do in clinical trials. Ultimately, we're trying to evaluate clinical benefit, meaning to evaluate whether that medical product has some positive impact on something that is really important to patients, such as how long they live or how they feel or function in daily life. And so I'm showing here the definition of clinical benefit, which is from the BEST glossary, and this is a living resource that was developed jointly by the FDA and the NIH to harmonize on concepts related to biomarkers, COAs, and other tools. So how do we assess clinical benefit? What are some of the tools that we have in our toolkit? Well, Dr. Leptak in his presentation discussed validated surrogate endpoints, but for the purpose of my presentation, I'll focus on COAs. COAs are measures that can be reported by patients themselves, clinicians, or observers. They can also include other measures such as performance outcome measures. How do we now determine which type of COA is best for our clinical trial or context of use? Well, patient-focused drug development doesn't necessarily mean we must always use PRO assessments. Rather, it means that we need to think about what is important to measure in a particular population, what is really clinically meaningful to clinicians, to patients, and other stakeholders, what can be potentially modified by a therapeutic or any type of intervention during a clinical trial, and then we can determine how best to measure it in a particular clinical trial. It may be that we are interested in assessing symptoms in that population, and the population can report for themselves, and in this case, we would use a PRO assessment. If clinical judgment is required to interpret an observation, then we turn to a clinician-reported outcome. If the outcome is observable and it is in daily life, then we may select an observer-reported outcome. And this is, in fact, what is very commonly used when our patients are young children or in patient populations who can't provide self-report. And in some cases, an actual demonstration of a defined task may be appropriate, and then we would use performance outcome measure. Last but not least, we're also seeing increasing interest in using digital health technologies that can passively collect continuous data from patients on important functions such as activity or sleep as they go about their daily lives. So for reference, this slide shows the definitions of the different types of COAs from the current version of the BEST glossary. And this resource also includes a discussion of each type, as well as some examples. Now I'd like to talk a little bit more about clinical benefit. We can think about clinical benefit in terms of direct evidence as well as indirect evidence. Direct evidence of clinical benefit is derived from studies with endpoints that measure survival or how patients feel and function in their lived experiences, while indirect evidence of clinical benefit is derived from studies with endpoints that measure things other than that are related to how patients survive, feel, or function. And so let's explore this concept a little bit more. And it might be helpful to think of in terms of a continuum of direct and indirect evidence of clinical benefit. Depending on how indirect something is, the more evidence we may need. So for example, let's take performance outcome measures such as timed walking tests or word recall tests. These measures are thought to be somewhat indirect because they are not measuring exactly how a patient feels or functions in their daily life, but instead they are intended to closely approximate patients' lived experience. And with these types of performance measures, it is critical to really understand what the performance test is actually measuring, what that score represents, and what all that means in terms of meaningful benefit to patients. Biomarker endpoints, which we see on the right side, are generally considered surrogates for clinical benefit and are at the far indirect spectrum, side of the spectrum. Now that we've discussed the important role of COAs, let's talk about the FDA review of COAs. Why is it important for FDA to evaluate our outcome assessments? And this really stems from our drug regulations, which highlight the need for well-defined and reliable assessment methods. In essence, our endpoints and assessment methods form the basis of labeling claims which cannot be false or misleading. Another important consideration is that the measurement should minimize variability or noise, and so that this can be sensitive to true change in a patient's condition. So now that we've discussed the why, how does the FDA review COAs for their adequacy to support regulatory decision-making? Importantly, FDA evaluates an instrument in the context of its intended use. In other words, in its context of use. And this includes things like the clinical trial and patient population where the COAs will be used, how they will be used, including whether they will be used to support labeling claims, and what claims are actually being targeted. In other words, there is no such thing as an instrument that has been validated for all purposes. The FDA Bureau Guidance of 2009 describes good measurement principles that are applicable to all COA types more broadly. So let's talk a little bit about the measurement properties that we review. We start with content validity, which is defined as the extent to which the content of an instrument represents important aspects of what it claims to measure, that is to say, the concept, in that intended use in patient population. This is, of course, key, as we want to ensure that the labeling is accurate with respect to the concept of measurement and is not misleading in any way. Content validity is supported by both qualitative and quantitative evidence. Importantly, if content validity evidence is inadequate, there is really little reason to test or consider other measurement properties. Other measurement properties that we review are shown here, and these are primarily quantitative in nature. They include reliability or reproducibility of the measure, such as test-retest reliability or inter-rater reliability, construct validity, which assesses whether the quantitative associations with other variables are as expected, and finally, ability to detect change. So what are some of the common COA review issues that we encounter? Well, one question that we ask is, was the instrument developed with input from the relevant stakeholders? If not, the risk is that it may omit what is most important and relevant. It may include irrelevant content, or its instructions, questions, and response options may not be well understood. And this can lead to noise in measurement, as well as lack of clarity on what we're actually measuring and whether that is in alignment with what is intended. Some other common COA review issues are shown here. Is the instrument appropriate for the study design, patient population, or research question? If not, the risk is that it may be poorly matched to the severity of the patient, so that the patient may be either at the low end of the scale or the high end of the scale. It may not be reliable, valid, or responsive to change. For example, use of a dexterity test developed for the general population wouldn't be appropriate for use in patients with visual impairment, for example. It may capture something important to patients, but not what the drug is targeting. The other thing is are the instrument's concept clear and well-defined? Is the content reflective of the concept that we're trying to measure? And if not, it may be very difficult or impossible to accurately describe what is measured in product labeling. So now I'd like to say a few words about seeking advice from the FDA. To meet the challenges of patient-focused outcome measurement and to ensure outcome assessments are fit for purpose for drug development, we recommend early consultation and close collaboration with the FDA throughout drug development. And we have 3 pathways I'd like to just highlight here for engagement in the Center for Drugs. First, of course, CDER routinely provides advice within individual drug development programs. And we encourage drug companies to begin discussions about their COAs as early as possible in drug development, including in the pre-IND stage, to ensure there's enough time to for the COA development and selection before the phase 3 clinical studies. The other 2 pathways I'm highlighting here are outside a particular drug development program. One is the Drug Development Tool Qualification Program, where we can work with stakeholders to create and qualify COAs, biomarkers, and other tools that meet unmet public health needs and can be used publicly across multiple drug development programs within the appropriate context of use. Another pathway is through various meetings, such as critical path innovation meetings, where stakeholders can have informal discussion and receive general non-binding feedback from the FDA on specific methodologies or tools, including COAs or biomarkers. And now I'd like to say just a few words about the drug development tool qualification pathway for seeking advice. A drug development tool, or DDT, is defined in statute as a biomarker, a COA, or any other method, material, or measure determined to aid drug development and regulatory review. The DDT qualification process, which was initiated approximately 10 years ago now, is codified as part of 21st Century Cures legislation and the process formalized. Qualification is simply a regulatory review process for drug development tools and is completely voluntary. If a tool has been qualified, it means that within the stated context of use, the tool can be relied upon to have a specific interpretation and application in drug development and regulatory review. The program allows us to qualify tools that will be publicly available for use in any drug development program in the qualified patient population and context of use without necessitating re-review by the agency. And there are 3 qualification programs currently, as shown here. With that, I thank you for your attention, and I'd like to leave you with some helpful resources.